Good morning, students. In today's class, students, we'll be studying about sexual reproduction in flowering plants. So basically here emphasis is given on angiosperms. Okay, so emphasis is given on angiosperms. Okay, so angiosperms are the flowering plants here. So here, students, we'll study about structure of the flower, the parts of the flower, the male parts, female parts, microsporogenesis, oogenesis, and after that we'll study the structure of gynoecium and all that, fertilization, and finally till the formation of fruit and different types of fruit. Okay, students. So we'll start with the class. Just a minute. We'll start with sexual reproduction in flowering plants. So all flowering plants, that is angiosperms, will show sexual reproduction. So flowers are special modified structures which are involved in sexual reproduction. Okay, so where does the flower arise from? The flower arises from the floral primordia. Okay, so several hormonal and structural changes will take place which will result in differentiation and development of floral primordia. Okay, this floral primordia will give rise to the flower. So inflorescence is the one that bears the floral buds and then the flowers. So inflorescence is basically the structure which will bear the floral buds and the flowers. Okay, so now a flower will have two major oils. So what do we call it as? Essential oils and non-essential oils. In the essential oils, it has two parts, andrecium and gynoecium. Andrecium and gynoecium. Non-essential oils include calyx and corolla. Calyx and corolla. Okay. So calyx and corolla. Calyx basically has individual units called sepals. Corolla has individual units called petals. Okay. Sepals ಮತ್ತು petals. So it is a male reproductive part of the flower. Okay. Andrecium. So now ಇಲ್ಲಿ ನಾನ್ ಎಸೆನ್ಷಿಯಲ್ ಓಲ್ಸ್ ಬಗ್ಗೆ ಅಷ್ಟು ಇಂಪಾರ್ಟೆನ್ಸ್ ಕೊಡೋದಿಲ್ಲ, ಯಾಕಂದ್ರೆ ಅದು ರೀಪ್ರೊಡಕ್ಷನ್ ಅಂದ್ರೆ ಇನ್ವಾಲ್ವ್ ಆಗೋದಿಲ್ಲ. ಬಟ್ ಎಸೆನ್ಷಿಯಲ್ ಓಲ್ಸ್ ಏನಿದೆ ಅದು ರೀಪ್ರೊಡಕ್ಷನ್ ಬೇಕೇ ಬೇಕು, ಅದರಿಂದ ಅದನ್ನ ಎಸೆನ್ಷಿಯಲ್ ಓಲ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಇಟ್ ಶುಡ್ ಬಿ ಈದರ್ ಮೇಲ್ ಆರ್ ಫಿಮೇಲ್ ಆರ್ ಬೋತ್. ಸೊ ಈದರ್ ಆಂಡ್ರಿಷಿಯಂ ಅಥವಾ ಗೈನೀಷಿಯಂ ಇರಲೇಬೇಕು ಅಥವಾ ಎರಡೂ ಇರಬೇಕು, ಅಥವಾ ಯಾವ್ದಾದ್ರು ಒಂದಾದ್ರೂ ಇರಲೇಬೇಕು. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ both andrecium and gynoecium may be present, or only andrecium or only gynoecium is present. So it is the male reproductive part of the flower, andrecium. So it consists of whorl of stamens. Whorl andre ಒಂದು circle of stamens. So their number and length are variable in different species. So within the same species, usually they are not variable, but among different species, their number is variable. Okay, so students, the diagram of the flower, okay, the diagram of the flower is very important for your theory exam. Okay, ಮತ್ತು parts ನಾವೀಗೇನು ಓದ್ತೀವಿ, that is very important for your NEET and CET exam. Okay, so take it down, students. ನೋಡ್ಕೊಳ್ಳಿದನ ಇದನ್ನ ಬರ್ಕೋಬೇಕು. So it consists of a whorl of stamens and their number and length are variable in different species. Same species ಅಲ್ಲಿ ವೇರಿಯಬಲ್ ಇರಲ್ಲ, ಆಲ್ಮೋಸ್ಟ್ ಸೇಮ್ ಇರುತ್ತೆ. Done. So ನೋಡಿ, what are the parts of the stamen? Stamen has two parts, one filament and one anther. Filament and a anther. Filament will basically help in attachment. Okay, filament will basically help in attachment to the thalamus or the petal of the flower. ನೋಡಿ, filament is long and stendal stalk. So ಇದು ಕಡ್ಡಿ ತರ ಇರುತ್ತೆ, which will help in attachment. So ಎಲ್ಲಿಗೆ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ? ಥಲಾಮಸ್ಗೆ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ or petalಗೆ ಅಟ್ಯಾಚ್ ಆಗುತ್ತೆ. If it is attached to the petal, then it is called epipetalous stamen. Take it down, students. Done. So if it is attached to the petal, then it is called epipetalous stamen. Then coming to anther. Anther is usually bilobed. That means it has two lobes. Okay, each lobe will have two theca. That means it is dithecous anther. So after often a longitudinal groove runs lengthwise separating the theca. The top to bottom there is a groove that is separating, which is called as dithecous anther. So there is a groove lengthwise that will separate the theca. So take it down. Okay. Done. So next we'll look at the transverse section, that is TS of a anther. So when the TS of an anther is taken, what do we see? It is tetragonal in structure. Okay, that means four corners, consisting of four microsporangia. Each one is located at each lobe. ನೋಡಿ ಇಲ್ಲಿ ಒಂದೊಂದು ತುದಿನಲ್ಲಿ ಒಂದೊಂದು lobe ಇದೆ ಇಲ್ಲಿ. Okay, so the microsporangia develops into pollen sacs. ಇಲ್ಲಿ ಏನಿದೆ, microsporangia will develop into pollen sacs and extends longitudinally through the length of the anther. So ಪೂರ್ತಿ ಲೆಂತ್ ಒಂದು microsporangia ಇರುತ್ತೆ. Okay, so it is packed with pollen grains. So take it down. Done. So we go to the next part. Nodi, if you take a cross section of the anther, okay, one microsporangia would be in each corner. So each microsporangia is usually circular in outline. You can see here, nodi, circle shape alli ide. It is a form of a circle here. Okay, this part. Now, each of this is surrounded by four wall layers from the outside. Which are those four layers? Epidermis, endothecium, middle layers, and tapetum okay. So the outer three layers, that is epidermis, endothelium and middle layer, they will help in protection and dehiscence of the anther, protections and dehiscence of the anther to release the pollen, whereas the inner tapetum will provide nourishment. Okay, so ಇದನ್ನ ಫಸ್ಟ್ ಬರ್ಕೊಳ್ಳಿ. ಡೆಹಿಸೆನ್ಸ್ ಅಂತಂದ್ರೆ ಏನು? ಆಂಥರ್ ಮೆಚೂರ್ ಆದಾಗ ಒಡೆದೋಗೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಡೆಹಿಸೆನ್ಸ್ ಆಫ್ ದಿ ಆಂಥರ್. ಸೊ ಟೇಕ್ ಇಟ್ ಡೌನ್. Then, so we go to the next part. So the tapetum is the innermost layer. Tapetum is the innermost layer which will nourish the developing pollen grains. So ಒಳಗಡೆ ಇರೋ ಏನು ಸ್ಪೋರೋಜೆನಸ್ ಟಿಶ್ಯೂ ಇರುತ್ತಲ್ಲ ಅದಕ್ಕೆ ಫುಡ್ ಬೇಕು. That will be given by this tapetum. Are you understanding? So the cells of the tapetum will contain dense cytoplasm, okay, and are generally more than one nucleus, and will have generally more than one nucleus. Okay, then in young anther, each microsporangium has sporogenous tissue at the center, which consists of compactly arranged homogeneous diploid cells. So sporogenous tissues ಮಧ್ಯದಲ್ಲಿ ಇರುತ್ತಲ್ಲ ಅದರಲ್ಲಿ diploid cells ಇರುತ್ತೆ. These diploid cells will undergo division to form microspores later. Okay, so take it down. ಜ್ಞಾಪಕ ಇಟ್ಕೋಬೇಕು, ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. And so remember this, the sporogenous tissue will undergo meiosis to form microspores. ನೋಡಿ ಇಲ್ಲಿ, as the anther develops, each sporogenous cell, microspore or pollen mother cell, will undergo meiotic division to form microspore tetrad. So ಒಂದು ಸೆಲ್ ಡಿವೈಡ್ ಆದ್ರೆ 4 ಸೆಲ್ ಆಗುತ್ತೆ, 4 ಸೆಲ್ಲು ಜೊತೆಲಿ ಇರುತ್ತೆ, ಅದಕ್ಕೆ ಮೈಕ್ರೋಸ್ಪೋರ್ ಟೆಟ್ರಾಡ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಟೆಟ್ರಾಡ್ 4. ಸೊ microspores are arranged in a cluster of 4 cells. Always ನೋಡಿ ಇಲ್ಲಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇಲ್ಲಿ ಕಾಣಿಸ್ತಾ ಇದೆಯಾ, 4 ಸೆಲ್ಗಳು ಇದು ಜೊತೆನಲ್ಲಿ ಅಂಟ್ಕೊಂಡಿರುತ್ತೆ. ಓಕೆ, so the formation of microspore from pollen mother cells to meiosis is called microsporogenesis. And so remember this. Next part, microsporogenesis. Nodi. As the anthers mature and dehydrate, so as they grow up and they dehydrate, what do you mean by dehydrate? That means when they lose water, okay, the microspore will disassociate from each other and develop into pollen grain. The microspore will disassociate, that means they'll separate and they'll be taken away in air. So each microsporangium contains thousands of pollen grains, and these are released with the dehiscence of anther. These are released with the dehiscence of anther. So take it down. This is very important, students. Now, we'll look at the pollen grain. How does a pollen grain look like? A pollen grain is not a male gamete, students. Always remember it is the male gametophyte. All of you remember sporophyte and gametophyte generation in plants. It is a male gametophyte. Okay. So it is generally spherical, 20 to 50 micrometer in diameter. Then cytoplasm is surrounded by a plasma membrane, just like normal cells, and the pollen grains will have two layered wall, outer exine, inner intine. Okay, the outer exine, what is there, is made up of a hard substance, very hard substance, which is resistant to which is highly resistant organic material to acid, alkalis, and to heat. Okay, so which is made up of a chemical called sporopollenin, which is made up of a chemical called sporopollenin. So the exine, okay, so the exine will have certain places where the sporopollenin is absent. Okay, so that region is called is called germ pore, through which the pollen tube comes out. Okay, so pollen grains are preserved as fossils in the presence of sporopollen. ಸೊ ತುಂಬಾ ವರ್ಷಗಳಾದರೂ ಹಾಗೆ ಇರುತ್ತೆ, ಯಾಕಂದ್ರೆ ಸ್ಪೋರೋಪೊಲಿನ್ ಏನು ಆಗೋದಕ್ಕೆ ಬಿಡೋದೇ ಇಲ್ಲ ಪೋಲನ್ಗೆ. ಸೊ ದ ಎಕ್ಸೈನ್ is will have different patterns and design, whereas the intine will have a thin and continuous wall made up of cellulose and pectin. Cellulose and pectin. So inner to it, it will have two cells, a vegetative cell. Okay, it is bigger and has abundant reserve food material and irregularly shaped nucleus, whereas the smaller cell is the generative cell, which floats in the cytoplasm of the vegetative cell. Okay, it is spindle shaped with dense cytoplasm and a nucleus. Okay, so take it down. So ಈ diagram ಅಷ್ಟೇ, diagram with notes can be asked for 5 marks. Okay, in the theory exam. Take it down, students. ಸೊ ಅದಕ್ಕೆ ನಾನು ಪ್ರತಿ ಎಕ್ಸ್ಪ್ಲನೇಷನ್ ಆದ್ಮೇಲೆ ಸ್ವಲ್ಪ ಟೈಮ್ ಕೊಡ್ತೀನಿ. ಸೊ ನೀವು ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್ಸ್ ಬರ್ಕೋಬೋದು ಅಂತ ಬರ್ಕೋಬೇಕು ನೀವು. Done. So now take this part, take down about sporopollen, what is it resistant to, and what are germ pores. ಈ ತರ ಕೀ ಪಾಯಿಂಟ್ಸ್ ನ ಬರ್ಕೋಬೇಕು. ಸೊ ನೀವು ನೀಟ್ ಓದಬೇಕಾದ್ರೆ ಇಡೀ ಟೆಕ್ಸ್ಟ್ ನ ಅಥವಾ ಇಡೀ ಮೆಟೀರಿಯಲ್ ನ ಓದಕ್ಕೆ ಹೋಗ್ಬಾರ್ದು. Just you should look at these key points and it should be easy for you to remember. So next, take down about intine. Intine, continuous layer made up of cellulose and pectin. Fast. Next one, put the two cells. So the larger cell is vegetative cell, the smaller cell is generative cell. Done. Next, see, majority of the pollen grains that are released are in two-celled stage. That means one generative cell and one vegetative cell. Whereas in others, the generative cell will divide mitotically to give two male gametes, and thus they are shed at three-celled stage. So three-celled stage, usually 40% of them, majority 60% of them are shed at two-cell stage. It is the generative cell. It is the generative cell which will divide mitotically to give two male gametes. That, so remember this, they usually shed at two cell stage, but in some cases, what will happen? The generative cell will divide mitotically to form two male gametes. So if somebody asks you which is the male gamete, it is the cells that are formed by the division of the generative cell. ಜ್ಞಾಪಕ ಇಟ್ಕೋಬೇಕು. Generative cells nucleus will divide into two to form male gametes, but the male gametophyte is the whole pollen. Gametophyte is the whole pollen. ಗೊತ್ತಾಗ್ತಿದೆಯಾ? This is very important. Okay. Now remember this. The shed pollen grains have to land on the stigma before they lose viability. ಅಂದ್ರೆ ಕರೆಕ್ಟ್ ಟೈಮ್ಗೆ ಅದೇನಾಗಬೇಕು? They should land on the stigma. If they don't land on the stigma, then their viability is lost. Waste. ಅದಾದ್ಮೇಲೆ pollen grain ಅದರದು activity ತೋರಿಸೋದೇ ಇಲ್ಲ. Okay. So remember that pollen grain will not show its activity. Then, so viability period of pollen grain is variable. It depends on temperature and humidity. Okay. So viability of pollen grains of some cereals, rice, wheat, etc. is 30 minutes. So some members of Leguminosae, Rosaceae, Solanaceae have viability for months. Okay. So take it down. So based on how good it is, the longer it will live. Done. So now, what is the economic importance of pollen grains? So the pollen grains are basically rich in nutrients, and these pollen tubes are used as food supplements, okay, as tablets and as syrup to increase the performance of athletes and race horses. And these pollen grains can be kept in material for longer period of time. Okay, so they're kept in pollen banks and for stored for years in liquid nitrogen. So the pollen grains of some plants, example parthenium or carrot grass, are allergic to some people and lead to chronic respiratory disorders like asthma and bronchitis. So take it down, students. Done. So next we go to the female part. Okay, we go to the female part, that is the gynoecium. Here it is the female reproductive part of the plant. Okay, if it has a single pistil, If it has a single pistil, then it is called as monocarpellary. Okay, if it has a single pistil, then it is called as monocarpellary. Okay, then if it has more than one pistil, then it is multicarpellary. So, okay, in multicarpellary, the pistils or the carpels might be fused. If they are fused, they are called syncarpous. If they are free, they are called as apocarpous. So, take it down. So, done. Next, each pistil will have three major parts: stigma, style, and ovary. Stigma, style, and ovary. Take it out. Stigma for receiving the pollen grains or a platform for the pollen grains. Style, elongated slender part through which the pollen tube will pass. Then we have the ovary, which is the basal bulged part of the gynoecium. Done. So next, the gynoecium inside the ovary is the ovarian cavity. Is the ovarian cavity or the locule in which placenta is located. In which placenta is located. From the placenta, the ovules will arise. Okay, that is the megasporangia will arise. So the number of ovules in an ovary may be one, as in wheat, paddy, mango, or else there might be many in the case of papaya, watermelon, orchids, etc. So take it down. The examples are very important. Done. So we go to the next part. So the ovule here, students, is attached to the placenta by a stalk called as funicle. So ಇಲ್ಲಿ ನಾವು ಉಲ್ಟಾ ಕಾಣಿಸುತ್ತೆ ಇದು, but the attachment might be straight here. Okay, so called as the funicle. So the junction between the body of the funicle, body of the ovule, and funicle is called as hilum. ನೋಡಿ ಇಲ್ಲಿ ಈ ಪಾರ್ಟ್ ಇದೆಯಲ್ಲ, this is the hilum. So each ovule has one or two protective envelopes which are called as integuments ಅಂತ ಹೇಳ್ತೀವಿ. So the integuments will protect the megasporangium inside. Okay, so at certain regions the integument will not close completely. So where there is a small opening called as the micropyle, opposite to the micropylar end is the chalaza, okay, which is the basal part of the ovary, Now enclosed within the integuments there is a mass of cells called as nucillus. This nucillus will have reserve food material for the embryo sac. So inside the nucillus is the embryo sac, that is the female gametophyte. So an ovule generally has a single embryo sac formed from a megaspore. So here the megaspore mother cell anta ಏನಿರುತ್ತೆ, okay, so that will undergo meiosis to form the megaspore. So we'll do that later. So first take down this ovule structure. ನೋಡ್ಕೊಳ್ಳಿ, only the ovule structure can be asked in your theory exam and also its description. Take it down. Hilum ಎಲ್ಲ ಇಲ್ಲಿ ಏನೇನು ಹೈಲೈಟ್ ಆಗಿದೆ ಅದನ್ನೆಲ್ಲ ಬರ್ಕೋಬೇಕು. Those are very very important. So after every slide I give a one minute time. So please take it down. ಬರ್ಕೋಬೇಕು ಅದನ್ನ. Done. So now we go to the next part. Take this down. Nucillus. So just like how in microsporangium we have tapitum, in the megasporangium we have nucillus. Okay, so neat ಏನು ಮಾಡ್ಬಿಡ್ತಾರೆ, analogy ತರ ಕೊಡ್ತಾರೆ. So microsporangium tapitum, megasporangium nucillus. ಆತರ microsporangium we have the pollen, megasporangium we have the embryo sac. ಆತರ Then, so next one, we go to Megasporogenesis. So which is the megaspore here? Megaspores are formed from megaspore mother cells. So before that, remember the students, embryo sac is the female gametophyte. So basically we'll study how the female gametophyte is formed. Okay. So here there is a single cell called as the megaspore mother cell. Okay, which will undergo meiosis to form megaspores. Okay, so the ovules are generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. So nucellus region ಅಲ್ಲಿ ಒಂದೇ ಒಂದು ಸೆಲ್ಲು ಮೆಗಾಸ್ಪೋರ್ ಆಗಿ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ. ಓಕೆ, ಸಿಂಗಲ್ ಮೆಗಾಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಆಗಿ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಒನ್ ಓಕೆ will have dense cytoplasm, prominent nucleus and reserve food material ಇರುತ್ತೆ ಸ್ವಲ್ಪ. Then it will undergo meiosis. So meiosis 1 ಆದಾಗ 2 ಸೆಲ್ ಆಗುತ್ತೆ, meiosis 2 ಆದಾಗ 4 ಸೆಲ್ ಆಗುತ್ತೆ. So totally 4 cells are formed at the end of meiosis. So what is it called as? It is called as a megaspore tetrad. It is called as a megaspore tetrad. Understand this, it is called as a megaspore tetrad. So this megaspore tetrad, there are 4 cells. Okay, out of these 4 cells, 3 cells will degenerate. So 4ರಲ್ಲಿ 3 cells will degenerate. Okay, and only one cell will survive, that is called as a functional megaspore, which will develop into a female gametophyte. Understand this, out of those 4, 3 of them will degenerate. Okay, and only one will survive at the end, which is called as the functional megaspore. So which develops into the female gametophyte, which develops into female gametophyte. So the embryo sac formation from a single megaspore is called monosporic development. Is called monosporic development. Okay, are you understanding? So first ಇದನ್ನು ಬರ್ಕೊಂಬಿಡಿ, ಫಸ್ಟ್ ಇದು ಬರ್ಕೊಳ್ಳಿ, take down this, then we'll go to the embryo sac. Done. So take this down. Done. So now look at this. Once the functional megaspore is there, okay, once the functional megaspore is there, because three of them have degenerated, okay, because three of them have degenerated, only one is remaining, only one is remaining. That one cell is the functional megaspore, which will mitotically divide to form two nuclei. So here the cell will not divide, but only the nucleus will divide. Okay, only the nucleus will divide. So once the nucleus divides, once the nucleus divides, it will keep dividing. So there'll be totally of 3 nuclear divisions to form 8 nuclei totally. First, the nuclei will divide into 2, 2 will divide to form 4, 4 will divide to form 8. So this is called as free nuclear divisions will take place. Okay, so that means the cell will not divide, but the nuclei will divide. Okay, so free nuclear divisions will take place. Okay, and totally how many nuclei are formed? 8 nuclei are formed. 8 nuclei are formed. So this is a 8 nucleate condition. Then after the 8 nucleate condition, what will happen? The cell walls are laid in between the 8 nuclei. The cell walls are laid in between the 8 nuclei. Okay, leading to formation of female gametophyte. Okay, leading to formation of the female gametophyte. Out of that, ನೋಡಿ 6 out of 8 nuclei are surrounded by cell walls and are organized into cells. So which are those? On the top, on the top we have three antipodals, three antipodals, and in the end we'll have two synergids and one egg cell and central cell. So first we'll look at the central cell. ನೋಡಿ ಇಲ್ಲಿ. The remaining two polar nuclei are situated in the egg apparatus in the large cell. Okay. Now, a typical mature embryo sac is 8 nucleated and 7 celled. 8 nucleated and 7 celled. So 3 cells are grouped at the micropylar end to form the egg apparatus. So which are those 3 cells? 2 synergids and 1 egg cell. 2 synergids and 1 egg cell. So synergids have special cellular thickenings, okay, at the micropylar tip and are called as filiform apparatus, and are called as filiform apparatus. These filiform apparatus will guide the pollen tube towards the egg. So ಯಾವಾಗ fertilization ಆಗುತ್ತೆ ಅವಾಗ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ synergids. Then 3 cells at the chalazal end, they are called as antipodals, and a large central cell which has 2 polar nuclei. So ನಿಮಗೆ 7 ಸೆಲ್ ಯಾವುದಾದ್ರು? 3 ಆಂಟಿಪೋಡಲ್ಸ್, 2 ಸೈನರ್ಜಿಡ್ಸ್, 5 ಆಯ್ತಾ, ಒಂದು ಎಗ್ 6 ಆಯ್ತಾ, ಮತ್ತೆ ಒಂದು ಸೆಂಟ್ರಲ್ ಸೆಲ್ 7. Okay, so totally 7 cell. So how 8 nuclei? All cells will have one nuclei. 7 nuclei ಆಗುತ್ತೆ, but one extra nuclei is found in the central cell. Okay, that is the polar nuclei. Okay, so now students, take this down. ಇಲ್ಲಿ ಬರ್ಕೊಳ್ಳಿ, ಇಲ್ಲಿಂದ ಬರ್ಕೊಳ್ಳಿ. How does 2 cells become 4? How does 4 become 8? I'll just give you a minute. Then take it down. Done, students. So take down the next one. ಈ ಪಾಯಿಂಟ್ಸ್ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟು, ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ. So take down, students, all of you. Next part, look at the students. Formation of female gametophyte overall, how it looks like. Look at the steps, please. So just a second, students. We'll go to the next part. Just a minute. So after the development of female and male parts, so usually we look at the fertilization parts. Okay, so if fertilization has to take place in plants, pollination has to take place. So what is pollination? It is a transfer of pollen grains from the anther to stigma. Okay, from anther to stigma of a pistil. So from it has to get transferred from anther to the stigma. So this is pollination. So we have different types of pollination based on the source of pollen. We have autogamy, then we have geitonogamy, and then we have xenogamy. Okay, so take down autogamy, geitonogamy, and xenogamy. So what is autogamy? Autogamy means self-pollination. So it is the transfer of pollen from anther to stigma of the same flower. So here in flowers which have exposed anthers, so usually autogamy is rare. But remember students, autogamy in such flowers requires synchrony of pollen grain release and stigma receptivity. ಮತ್ತಂದ್ರೆ ಏನು pollen grain ರಿಲೀಸ್ ಆಗೋದಕ್ಕೂ ಸ್ಟಿಗ್ಮಾ ರಿಸೀವ್ ಮಾಡ್ಕೊಳ್ಳೋದಕ್ಕೂ ಎರಡು ಒಂದೇ ಟೈಮಿಂಗ್ ಅಲ್ಲಿ ಆಗಬೇಕು. ಸೊ ಫಸ್ಟ್ ಪೋಲನ್ ಗ್ರೈನ್ ಡೆವಲಪ್ ಆಗ್ಬಿಡುತ್ತೆ, ಆಮೇಲೆ ಸ್ಟಿಗ್ಮಾ ಡೆವಲಪ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ ಆಗಲ್ಲ, ಎರಡೂ ಒಟ್ಟಿಗೆ ಆಗಬೇಕು. ಓಕೆ, so and they should be very close to each other, and they should be very close to each other. So take it down. Done. So we go to the next part. ನೋಡಿ ಏನಾಗುತ್ತೆ. So here how autogamy is. Autogamy, okay, can take place by two types of flowers. So first one is chasmogamous flowers. First one is chasmogamous flowers. Second one is cleistogamous flowers. Chasmogamous flowers are those flowers where flowers with exposed anthers and stigma, ಅಂದ್ರೆ ಓಪನ್ ಆಗಿ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಆಗುತ್ತೆ. Cleistogamous flowers are those which do not open at all. So first ಇದನ್ನು ಬರ್ಕೊಳ್ಳಿ, and also take down the examples. Done. So next part ಹೋಗೋಣ, chasmogamous ಬರ್ಕೊಳ್ಳಿ. Done. Next one, cleistogamous flowers. So they do not open at all. Cleistogamous flowers are those which do not open at all. Okay, so the anther and stigma will lie very close to each other. So their pollination will take place even before the flower opens up in the bud stage itself. So when the anthers dehisce in the flower buds, the pollen grains will come in contact with the stigma for pollination. So cleistogamous flowers produce assured seed set even in the absence of pollinators. So cleistogamy leads to inbreeding depression. So continuous inbreeding will lead to sterility and lessening of yield, which is called as inbreeding depression. So take it down. Done. So we go to the next part, gytinogamy. So here it is a transfer of pollen grains from anther to stigma of another flower, but of the same plant. ಒಂದೇ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಬೇರೆ ಹೂವು. ಆದರೆ ಇದು functionally it is cross pollination involving pollinating agents, because it has to go from one flower to the other flower of the same plant, but it is genetically autogamy. Why? Because it is of the same plant. Okay, so take it down. Gytenogamy. Done. So next we go to xenogamy. Here it is a transfer of pollen between two different flowers from two different flowers from two different plants. So it brings about genetically different pollen grains to the stigma. Xenogamy. So here pollinating agents are required. So next one, we look at pollination. So there are different agents of pollination. Pollination. Okay, those are abiotic agents and biotic agents. So those are abiotic agents and biotic agents. So remember students, abiotic agents and biotic agents. Okay. So abiotic agents are wind and water, whereas biotic agents are insects, birds, primates, arboreal, rodents. Okay. All these are biotic agents. So first we'll look at abiotic agents. Abiotic agents include wind and water. First, we'll look at wind. So, pollination by wind is called as anemophily. So, it is the most common biotic agents. Okay, they are the most common biotic agents. So, wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. So, why? Because they should be able to pick up pollen very easily and spread pollen also. And the stigma are long and feathery so that they should trap pollen grains, and it is very quite common in grasses. So how are these pollens so effective? So they produce large number of pollens. Okay, they produce large number of pollens. Then the pollen grains are light and sticky. Light and sticky. Remember this, they are light and non-sticky. Sorry, non-sticky. They often possess well-exposed stamens so that easy dispersal of pollen grains will take place. Large and feathery stigma to trap airborne pollen. Okay, so remember the students. So in Berkeley, it is called as anemophily. So take this down. What are the ways of effective pollination, or what are the characteristics of airborne pollen? Done. So we go to the next part. Here, pollination by water is called as hydrophily. So it is seen only in aquatic plants. Example, Vallisneria, Hydrilla, Zostera and seagrasses. So it is usually rare and is limited to only about 30 genera. Okay, mostly monocotyledonous plants. So here students, this is very important. So but in lower plants, water is a regular mode of transport of water gametes, especially in the case of bryophytes, Okay, so distribution of some bryophytes and pteridophytes is limited because they need water for transport of male and female gametes for fertilization. So what happens in Vallisneria students? In Vallisneria, the female flower reaches to the surface of water along with a long stalk, and the male flowers of the pollen grains are released onto the surface of water, and they are carried away by water current, and they reach the female flowers So in sea grasses, so female flowers will remain submerged in water. The pollen grains are long and ribbon like, and they are carried by water currents, and they reach the stigma. Okay, so next note, pollen grains of most of the water pollinated species have a mucilaginous covering to protect them from wetting. So usually if it is dry, normal sporopollen irutte ಆಗುತ್ತೆ because it's in air, but here it is in water. So not all aquatic plants will use hydrophily. Why? Because some of them that are exposed outside water, they don't require hydrophily. So there it is either entomophily or anemophily. Okay, so take this down. So 30 genera, Vallisneria, ಇದನ್ನೆಲ್ಲ ಬರ್ಕೊಡಿ. Done. So we go to the next part. So take down about Vallisneria and seagrasses. Take this down. Then we'll look at biotic agents now. The biotic agents include various types of organisms. So usually they pollinate a large variety of flowering plants. Example, bees, butterflies, okay, beetles, wasps, ants, moths, all of them. Okay, here we have specific names for certain types of pollinating agents. So pollination by insects is called entomophily. Okay, so take down. Often flowers or animal pollinated plants are specially adapted for particular type of species of animals. So first take down this and I'll give you different types of names for pollination. ಇಲ್ಲಿ ಒಂದೇ ಒಂದು ಕೊಟ್ಟಿದ್ದಾರೆ ಅಷ್ಟೇ, ಎಂಟಮೋಫಿಲಿ. So I'll give you other names also. Take it down fast. Done. So now take down, students. Pollination by birds is called ornithophily. Pollination by birds is called ornithophily. Okay. By insects, it is entomophily. Okay. By bats, it is called chiroptophily. By bats, it is called chiroptophily. By bats, it is called chiroptophily. Okay. Then by snails, it is called malacophily. Malocophily. Okay. Then myrmecophily. MYR MECOPHILY. Myrmecophily. Pollination by ants. Done. So these are the different types of biotic agents. Now, what are the special features of the flowers so that they are pollinated by insects? So they're large and colorful, fragrant and rich in nectar. Fragrant and sweet smelling antartha. So that the insects will get attracted to it. And the floral rewards kodutte idu. So that the insect will get attracted to it. Okay. Now, small flowers from inflorescence to make them visible. So when they come and sit, so there is easily pollination when the insects come and sit on them. Then flowers are pollinated by flies and beetles secrete foul odors to attract these animals. So ಚೆನ್ನಾಗಿರೋ ಸ್ಮೆಲ್ಲು ಅಷ್ಟೇ ಅಲ್ಲ, ಕೆಟ್ಟ ಸ್ಮೆಲ್ಲು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ, ಯಾಕಂದ್ರೆ moths and other ones should be attracted. So the pollen grains are generally sticky because they have to stick to the body of the insect. Okay, so this is very, very important, students. So when the animal comes in contact with the anther and stigma, its body gets the pollen grain. So when it comes in contact with stigma, this results in pollination. So take it down. Done. So take this down. So look at this. Some plants provide safe places as floral rewards to lay eggs. Example in the case of Amorphophallus. Okay, a moth species and plant yucca cannot complete their life cycle without each other. So both are dependent on each other. The moth will deposit its egg in the locule of the ovary, and the flower will get pollinated by the moth. So the larvae will come out of the eggs as seeds and will start developing. Take it down. This is a very important as symbiotic agent also. Done. So we go to the next one. ನೋಡಿ ಇಲ್ಲಿ, many insects will consume pollen and nectar without bringing about pollination. These are called as pollen or nectar robbers. ಸೊ ಏನು ರಿವಾರ್ಡ್ಸ್ ಇರುತ್ತೆ ಅದನ್ನು ತಗೊಳುತ್ತೆ, ಬಟ್ ದೇ ವಿಲ್ ನಾಟ್ ಡೂ ಎನಿ ಪಾಲಿನೇಷನ್ಸ್. ದೋಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ pollen or nectar robbers. ನೆಕ್ಸ್ಟ್ ಔಟ್ಬ್ರೀಡಿಂಗ್ ಡಿವೈಸಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ವಾಟ್ ಹ್ಯಾಪನ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಬಿಕಾಸ್ ಆಫ್ ಕಂಟಿನ್ಯೂಸ್ ಇನ್ಬ್ರೀಡಿಂಗ್, ಸೊ ಏನು ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಆಗಿ ಆಗಿ ಏನಾಗುತ್ತೆ, ಈಲ್ಡ್ ಮತ್ತು ಪ್ರೊಡಕ್ಟಿವಿಟಿ ಕಮ್ಮಿ ಆಗಿಬಿಡುತ್ತೆ, ಫರ್ಟಿಲಿಟಿ ಕಮ್ಮಿ ಆಗಿರುತ್ತೆ. ಸೊ ದೇ ವಾಂಟ್ outbreeding to happen. So plants will devise certain outbreeding devices, okay, so that only cross pollination takes place. Let us look at a few. Hermaphrodite flowers can undergo self pollination or autogamy, okay. So continued self pollination will lead in inbreeding depression. So to prevent this, they encourage cross pollination. So how do they do it? ನೋಡಿ ಇಲ್ಲಿ ಒಂದೊಂದಾಗಿ ನೋಡೋಣ. ಅವಾಯ್ಡಿಂಗ್ ಸಿಂಕ್ರನೈಸೇಷನ್. ಸೊ ಪೋಲನ್ ಮತ್ತು ಸ್ಟಿಗ್ಮಾ ಎರಡು ಒಂದೇ ಟೈಮ್ನಲ್ಲಿ ರಿಸೆಪ್ಟಿವ್ ಆಗೋದಿಲ್ಲ. Pollen will come out at some point, stigma will come out in some points. Are you understanding? Okay. Then arrangement of anther and stigma at different positions. So if the anther is below and the stigma is above it, so usually the chance of pollination will be less. Then self incompatibility. So it is a genetic mechanism to prevent self pollen. That means pollen of the same flower falling on the stigma of the same flower and from fertilization by inhibiting pollen germination or pollen tube growth in the pistil. ಅರ್ಥ ಆಗ್ತಾ ಇದೆಯಾ? ಸೊ ಇಲ್ಲಿ ಏನಾಗ್ತಿದೆ, ಸೊ ಒಂದು ಫ್ಲವರ್ ಅಲ್ಲಿ ಇರುವಂತ ಪೋಲನ್ ಅದೇ ಫ್ಲವರ್ ಇರುವಂತ ಸ್ಟಿಗ್ಮಾ ಮೇಲೆ ಬಿದ್ರೆ ಅಲ್ಲಿ ಅದು ಜರ್ಮಿನೇಟ್ ಆಗೋದಿಲ್ಲ. ಅದನ್ನ ಸೆಲ್ಫ್ ಇನ್ಕಾಂಪ್ಯಾಟಬಿಲಿಟಿ ಅಂತ ಹೇಳ್ತೀವಿ. ನೆಕ್ಸ್ಟ್ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್. ದಟ್ ಮೀನ್ಸ್ the male and female flowers produced on the same plant, okay, they will produce only male flowers or only female flowers, okay, only male flowers or only female flowers. Understand this, so that it prevents autogamy but not gytenogamy. Then last one, in dioecious plants, papaya male and female flowers are present on different plants, which will prevent both autogamy and gytenogamy. Okay, so we'll take this down. Done. Take down the third one. Self incompatibility. Then take down production of unisexual flowers. So examples are very important here. Done. So we go to the next one, unisexual flowers. Done. So now pollen pistil interaction. So remember students, when the pollen is released, it need not always fall on the stigma of the same species and the same variety. Okay. Sometimes incompatible pollen will also fall on the stigma. So only the pollen grain which is compatible should germinate. The, that is very important. Okay. So how do the plants will recognize whether it is a compatible or incompatible pollen? Based on chemical components produced by them. So if the pollen is compatible, that is of the right type, okay, its pistil will accept it and promotes post pollination events. Okay, so the pollen grain will germinate on the stigma to produce a pollen tube. The pollen tube will pierce through, will come out through the pollen tube, pollen germ pore, and will pierce through the stigma, style, and then will reach the ovary. Okay, now, so if the pollen is incompatible or of the right or the wrong type, the pistil will reject the pollen by preventing pollen germination on the pollen tube growth. So in plants which shed pollen grains at two-celled condition, that is vegetative and generative cell. So the generative cell will divide into two male gametes during pollen tube growth. So remember that generative cell is the one that will actually produce the male gamete and not the vegetative cell and not the vegetative cell. So all of you take it down, students. Done. So we go to the next part. So in plants which are said at three-cell condition, the pollen tube will carry two male gametes from the beginning, and it will pass through the pollen tube and reach the ovary. So when it reaches the ovary, okay, it will enter the ovule through the micropyle, okay, and then enter the synergids. In the synergids, they have filiform apparatus. They have something called as filiform apparatus. The filiform apparatus will guide the pollen tube towards the egg. Okay, will guide the pollen tube towards the egg. So ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ, filiform apparatus will guide the pollen tube towards the egg. Then what will happen? So this will move through that and fertilize. Nodiri, in crop improvement program in which desired pollen grains are used for pollination. So ಇಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ? So we cross two different plants based on our wish. So we do two steps here: emasculation and bagging. What is emasculation? It is the removal of anthers, okay, from the bisexual flowers before the anther dehisces. So why do we do emasculation? To prevent self-pollination. Then we bag it. Why? Because to prevent contamination from unwanted pollen. So ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ. Then mature pollen grains are taken from some other plant, and then these mature pollen grains are dusted onto the stigma and rebagged, okay, and rebagged and allowed to develop as fruits. So take it down. Emasculation and bagging. Done. So next one. For unisexual flowers, there is no need for emasculation because ಅದರಲ್ಲಿ ಮೊದಲೇ male flowers ಇರೋದಿಲ್ಲ. Okay, so easily you can take only from the male flowers, then spread it on. So next, double fertilization ಬರೋಣ. Once the pollen tube will enter the synergids, it will release its male gametes there. So after entering one of the synergids, the pollen tube will release two male gametes into the cytoplasm of the synergid. So one male gamete will move towards the egg cell and fuses with the nucleus to form a zygote, which is a diploid zygote. And the other one will move towards the two polar nuclei, and all three of them will fuse to form a triploid primary endosperm nucleus. As it involves the fusion of three haploid nucleus, it is called as triple fusion. Triple fusion. So take it down. Done. So take it down, students. This is very, very important. So take this down. Since two fusions are there, singamy and triple fusion. That is why it is called double fertilization. So this is an event which will take place only in angiosperm, students. So remember, it will take place only in the angiosperms. The central cell after triple fusion will become primary endosperm cell and will develop into endosperms. The zygote develops into a embryo. So take it down fast. Done, students. So we'll go to the next one. Post-fertilization structures and events. So what all will take place after fertilization? Endosperm development, embryo development, ovule into seeds, ovary into fruits. So you should know what is the fate of each one that will take place. So take down, students. Done. So let us look at each part. Okay. Endosperm development. So the primary endosperm cell will divide continuously to form triploid endosperm tissues. So which will have reserve food material which will help in the nutrition of the developing embryo. So this is very, very important. Okay. Done. So next part we go to. So here the primary endosperm nucleus will undergo successive nuclear division to give free nuclei. This stage is called as free nuclear endosperm, students. This stage is called as free nuclear endosperm. So the number of free nuclei will vary greatly. So the endosperm will become cellular due to cell wall formation. So students, the endosperm that you see in coconut, so ನಾವು ಏನು ತಿಂತೀವಲ್ಲ ತೆಂಗಿನಕಾಯಲ್ಲಿ ಅದು ಸೆಲ್ಯುಲಾರ್ ಎಂಡೋಸ್ಪರ್ಮ್, ಮತ್ತು ನಾವೇನು ಕುಡಿತೀವಲ್ಲ ನೀರು, ಎಳ್ನೀರು, ಅದು ನ್ಯೂಕ್ಲಿಯರ್ ಎಂಡೋಸ್ಪರ್ಮ್. ಜ್ಞಾಪಕ ಇಟ್ಕೋಬೇಕು. ಓಕೆ, ಕೀಪ್ ದಟ್ ಇನ್ ಮೈಂಡ್, ದಿಸ್ ಇಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಡನ್. ಸೊ ನಾವು ವಿ ಗೋ ಟು ದ ನೆಕ್ಸ್ಟ್ ಪಾರ್ಟ್. ನೋಡಿ ಇಲ್ಲಿ ಟೆಂಡರ್ ಕೋಕೋನಟ್ ಎಳ್ಳಿರುತ್ತಲ್ಲ, ಅದರಲ್ಲಿ ತಿಂತೀವಲ್ಲ ಅದು ಸೆಲ್ಯುಲಾರ್ ಎಂಡೋಸ್ಪರ್ಮ್, ಕುಡಿತೀವಲ್ಲ ಅದು ನ್ಯೂಕ್ಲಿಯರ್ ಎಂಡೋಸ್ಪರ್ಮ್. Done. Then embryo development. Nodi. The embryo will develop at the micropylar end, and the zygote will keep on dividing to form an embryo. So the formation of embryo is called embryogeny. So zygote will form a pro-embryo, then a globular embryo, then a heart-shaped embryo, and finally a mature embryo. So first we'll look at different types of embryos. First we'll look at dicot embryo. A dicot embryo will have an embryonal axis, okay, and two cotyledons. So the portions above the embryonal axis is called epicotyle, which terminates with plumule, that is the stem tip. The region below the embryonal axis is called hypocotyle, and its tip will have a root tip, which is covered by a root tip. That is the radical is covered by the root tip, okay, and this root tip is covered by a root cap. Okay, this is about a dicot embryo. Okay, take it down, students. Very important. So next we go to monocot embryo. A monocotyledonous embryo, okay, has only one cotyledon. So usually they have a lateral embryonal axis, and the lower end of the embryonal axis is the radical, and above it is the plumule. So the plumule is covered over by a coleoptile, and the radical is covered over by coleorhiza, undifferentiated sheath called coleorhiza. So the portion of the embryonal axis above the level of the attachment of scutellum is the epicotyle, and it has a shoot apex and a few leaf primordia, which is covered by coleoptile. It's covered by coleoptile. So take it down fast. Now we'll go to the next part. Seed. So what is a seed? Seed is the fertilized ovule that is found inside the fruits. Seed is the ovule, fertilized ovule found inside the fruits. So it is the final product of sexual reproduction. It is the final product of sexual reproduction. So it consists of seed coats, cotyledons, and embryonal axis. Seed coat, cotyledons, and embryonal axis. So the cotyledons are simple, generally thick and swollen due to storage of food, as in legumes. As in legumes. Done. So ನೋಡಿ ಇಲ್ಲಿ. ಸೊ ಕೆಲವರಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಸೊ ದ ಆಲ್ಬುಮಿನ್ ಸೀಡ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ, ವೇರ್ ದಿ embryonal development, primary endosperm is still retained. So whereas in some the endosperm is not retained, those are called as non-albuminous seeds. Normal pea, groundnut idu. Castor, ಎಳ್ಳೆಣ್ಣೆ ಬರುತ್ತಲ್ಲ ಅದು, it's a common example for albuminous seeds. Now as the seed matures, it will dry and it will have only 10 to 15% of moisture by mass. So the metabolic activity of the embryo slows down and it enters a stage of inactivity called dormancy. So under favorable conditions, moisture, O2 and suitable temperature, they will germinate. So since pollination, what are the advantages? So since pollination and fertilization are independent and water, seed and formation are more dependable. So dispersal into new habit takes place and they have reserve food material. Okay, reserve food material. Okay, next we go to fruits. So what are fruits? The ovary will develop into a fruit and the transformation of ovules into seeds, ovary into fruit will proceed simultaneously. So remember this, students, this is very important. So why? Nodili, advantages of seeds nodi. So they have a hard seed coat that will protect the young embryo. Then products of sexual reproduction, they generate new genetic combination and variation. Dehydration and dormancy of mature seeds are crucial for storage of seeds, and it can be used as food throughout the year and also raise the crop for the next season. Okay, ishtu advantages of seeds. Okay, so we'll stop here, students. We'll continue in the next class. So in the next class, we'll look at other forms of seeds. Then we'll look at fruits, parthenocarpy and all that, and we'll solve MCQs. So when you come for the next class, students, so please read content of this chapter, that is sexual reproduction in flowering plants. ಗೊತ್ತಾಯ್ತಾ? ಅದರ ಕಂಟೆಂಟ್ ಓದ್ಕೊಂಬನ್ನಿ. ಸೊ MCQ ಸಾಲ್ವ್ ಮಾಡೋದಕ್ಕೆ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ. ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು ಸ್ಟೂಡೆಂಟ್ಸ್. ವಿಲ್ ಎಂಡ್ ದಿ ಸೆಷನ್ ನೌ.